So the drug's been developed and it's been going into the animal experimentation phase. And if we get promising data from that animal experimentations, so we need to submit that data to the FDA, and FDA provides a permission for introduction of that drug into the human subject, and from that the clinical trial phase starts. So that procedure and the permissions which we get from the FDA, that is called as for investigational new drug. So are introducing an IND into the human subjects, that is called as investigational new drug. So this clinical trials is majorly divided into 4 phases, and during those 4 phases, you need to follow the ethical guidelines. Good clinical practice guidelines, which is being developed as an international conference on harmonizations, and different countries have also developed their guidelines of doing clinical trials in that particular countries. In India, for clinical trials, the guidelines for research is been laid down in Schedule 5 of Drug and Cosmetic Rules. In Drug and Cosmetic Rules, there are different schedules. You can go through that schedules online, and it's also having Schedule Y in that. That Schedule Y basically states requirements and regulations for conducting clinical trials on human subjects in India. So, according to that guidelines, you need to follow them. Now, there are 4 basic phases of clinical trial. First phase is called as human pharmacology and safety. Important word is safety here, where your main aim is to check whether the product is safe or not. In phase 2, now you are going for therapeutic exploration, how efficacious that product is, and its pharmacological or pharmacokinetic profile and dose ranging. In phase 3, now you are going into the larger number, and now you are confirming those studies of efficacy, safety, and pharmacokinetic profile through therapeutic confirmation and comparisons. And then you get marketing permission if the results are appropriate. And after the marketing, you are introducing drug into the market, into the real world, and again you need to check periodic data for long time side effects, and that is called as post marketing surveillance or studies. So 4 major phases. Now in phase 1, actually limited number of participant gets included, for example 20 to 80 participants up to several months, and our main aim is to check safety of that medications. So these things mostly been done on healthy volunteers. Okay, there are sometimes patients also there, but mostly it's been done on healthy volunteers, these phase 1 studies. Now, if we get promising result, the drug enters into the phase 2 study where the participant number almost doubles. So here 100 to 300 participant gets involved, and the main aim is to check the efficacy of a drug, how efficacious. So here we are introducing this drug into the patients actually. So patients are involved here. We are introducing drugs and we check for efficacy, how efficacious that drug is, and we also do some dose ranging studies. So dose ranging is also been done, like this is the dose, this is the dose range. Below this dose range there won't be any therapeutic effect. Above this dose range there are chances of adverse drug reaction. So all these things been collected in this phase 2. Then comes the phase 3 where the participant size increases, where even 1000 to 3000 participant gets involved, and it takes for longer time. Here that can be a multi-centric as well, right? So different centers, different countries, different ethnic groups been chosen for that studies. So depending upon the country's guidelines of clinical trials, this kind of phase 3 studies been done as a multi-centric trials, and it actually eventually checks or confirms the therapeutic efficacy of that phase 2 trial. So it's again efficacy, safety, and dosing study. It's more of an confirmatory studies because you are doing larger populations of different ethnicities. and then it goes, if the data are quite promising, then their drug goes into the FDA for permission into the real world. And if FDA permits, then it goes into phase 4, that is called as post marketing surveillance, where it's been introduced into many patients in real life, and from the clinicians the data has been recorded about the safety and efficacy, but most important is for long-term effectiveness and long-term side effects, because here you have done studies for shorter time, right? But you don't know what happens after longer time. For an example, some patients develops carcinoma after 10 years of taking drugs. So these kind of things you have not identified in this phase 1, 2 or 3. So this can be identified on phase 4 only, and you will be surprised there are fewer drugs which produces this kind of long-term side effects. Like for an example, diethylstilbestrol, it's a one kind of a hormonal medicines, and if a woman takes her daughter or her female child develops vaginal cancer, it's a huge time. So this kind of example shows that long-term efficacy and safety data are very much important, and that can be identified through this kind of phase 4 studies, where periodically safety and efficacy data needs to get submitted to FDA by particular pharmaceutical company. So these are 4 major phases of clinical trials. Now recently, one more phase which been developed in this clinical trial is called phase 0. It is also called as microdosing. Now what is it? It's actually a newer strategy. Let's talk about it. It's very interesting. So why this new strategy is being developed? It's been developed to reduce the cost and the time of drug development process. So what is the strategy? The strategy is before the phase 1 trial. So before the phase 1 trial, you make this kind of phase 0 trial, where in healthy volunteers you introduce drug, but how much? About 1/100th time of drug, or 100 microgram of the total dose. So it's a very small number in comparison to therapeutic dose. So in extremely small amount, a dose of a drug's been introduced into the healthy volunteer. For what? For checking the pharmacokinetics. So to check the pharmacokinetic profile, you have various newer mass spectrometry instruments are available, and through them you can check the ultra low levels of drugs, and you can get the pharmacokinetic profile. So what is the advantage of doing this kind of stuff? The advantage is, because of that such a low profile of dose, you not expect any kind of adverse drug reactions or toxic reaction in these healthy volunteers. So there is no issue of any harm in these healthy volunteers, because you are introducing extremely low amount of drugs. But the major advantage here would be, it will cancel the phases of the animal pharmacokinetic studies and even phase 1 human trials, because we are doing on animal also pharmacokinetic studies, and we are doing phase 1 studies to know the safety profile and pharmacokinetic. So all these things would be elaborate the needs. So all these things would be cancelled if we go through that phase 0 trials. So it will obviate the need for animal pharmacokinetic studies, and because of this, the time and the cost and resources would be saved, and that can be helpful in this clinical trial to the pharmaceutical companies. So this is how a new phase of clinical trial has been there, that is phase 0 or the microdosing studies.